Well, I'm Filippo Crea, Editor-in-Chief of the European Heart Journal, and it is a great pleasure to stay with you today for our monthly dialogues. Dialogue. You know very well that we have these monthly dialogues who are free and live streamed, and they are Socratic conversation among editors, authors, and readers of the journal. Today is a very exciting dialogue. It is my privilege to ask Professor Perry Elliott, Executive Editor of the European Heart Journal, to start the dialogue with a famous guest, Professor Silvia Priori from Pavia. And the title of this dialogue is Precision Medicine in the Prevention of Sudden Death, of Sudden Cardiac Death, Past, Present, and Future. Perry and Silvia. Please, Perry, you can start.
Thank you very much, Filippo, and it's indeed a great honor to be able to present our guest today, Professor Silvia Priori. Silvia is originally from Torino in Italy, but qualified in cardiology and cardiovascular pathology in the University of Milan. She then undertook her postdoctoral training, first of all in the United States at the University of Washington, St. Louis, and then back in Italy at the Telethon Center. She became a full professor of cardiology very recently, but started as an associate professor in Pavia in 2001, I believe, which is where she formed her clinical and research groups and began her journey into the field of inherited arrhythmias and sudden cardiac death. Silvia, I think, is the epitome of an international scientist. She's worked in New York as a professor of medicine at NYU. She's director of the molecular lab at CINEC in Madrid, and she's been honored by august bodies all over the world, including the Heart Rhythm Society, the American Heart Association, and of course the European Society of Cardiology, where she has received the gold medal for basic science. Silvia, complimenti, a fantastic career, and it's a great privilege to have you here this evening with us.
Thank you very much, Perry.
So Silvia, I suppose we should start with a little bit of a retrospective. And so where did your journey into the whole field of inherited arrhythmias and sudden cardiac death begin?
That's an interesting question indeed, because it started quite early. I was in the fourth year of medicine in Milano, and I was convinced that my life would have been that of a neurologist. And I was following with passion the clinic of inherited skeletal muscle diseases. At that point, I started fainting for unexplained reasons. So the first time, the second time, then I had a neurological workup that was completely negative. And then they said, well, you know, a female fainting unexpectedly, maybe there is something that those guys in cardiology are doing, and referred me to Peter Schwartz as a patient. So my door of entry has been as a patient. Luckily enough, I didn't have long QT syndrome, but I found the passion of my life. So
what a fantastic way to start. You've seen it from both sides of the desk, exactly. Then, both as a patient and a doctor. I mean, Silvia, you've written, I think, more than 350 papers, and you've made some really seminal contributions to the field. But if you look back, what do you think has, well, I suppose, first of all, been the most important contribution to science? And what is the one that pleases you the most? What is the one that you're most personally proud of?
Yeah, the one that I'm passionate about has been the discovery of the role of the ryanodine receptor in arrhythmias, because it goes beyond having found a gene for a disease that is important, but there are so many genes responsible for diseases. I think that the importance of that discovery has been to create a new reasons for basic science understanding of the role of intracellular calcium in arrhythmogenesis. And so most of the pleasure in that discovery has been to see that all of a sudden more people got interested because there was a translational potential development on what they were discovering in the ryanodine receptor. And I'm still seeing with a great pleasure, you know, when people ask me for our original animal model, because there is still so much to discover in this field that is great to feel that you made an observation and that has stimulated other scientists to move on.
So that's the scientific one. Is that genuinely your most favorite paper or your contribution, or do you have
Yes,
a secret one too?
No, I mean, it is definitely one of the paper that I like, because I remember a lot of stories around then that, that we discovered it in August, so was the time of going on vacation with the family. I didn't want to go on vacation. I sent the samples to Andy Marks for interacting with him on the ryanodine receptor. And so my mind was not on vacation that summer. So is a point important in my life. Now I have sort of departed the preference, because I'm so intensively with my teams involved in the gene therapy development, that the 2012 paper by Marco De Negri as a first author on the therapy of calsequestrin has become the one that is important for me at this point of the development of the research.
Okay. I mean, in one of the themes this evening is personalized medicine. And in a way, what you do is the ultimate in personalized medicine. It's identifying not only a gene, but an individual mutation that one could potentially target. But do you think that cardiovascular medicine is ready for personalized medicine? Can it actually embrace it?
I think that there is a big diversity among the diseases. If you take it from the perspective of long QT syndrome, where really the three genes that are not many for a disease with a prevalence of 1 in 2,500, so not very rare, that 80–90% of the patients can be now genotyped with mutations that have a clinical significance, and we can re-stratify, target the therapy. So if I have to think in cardiovascular, an area in which we have personalized medicine at a reasonable level of calling it personalized approach is the long QT syndrome. So we are lucky in that respect. Then if you look from the perspective of Brugada syndrome, we are very far away from that, with a lot of fundamental understanding of the disease that still has to be done. But in general, in the field of cardiology, big progresses are being made also in the genetic of polygenic diseases, or definitely not Mendelian conditions. And so that is the part that is very exciting of the most recent approaches and discovery, especially in ischemic heart disease, where certain loci have been confirmed over and over, and they are, I would say, ready to go into the clinic.
So, I mean, your passion for this is evident, but you've also been responsible for the Congress program for the ESC. And I know that you have this challenge, you've had this challenge each year of trying to bring more science to this, a group, thousands of type A cardiologists who think about not molecular diagnoses, but they think about stents, and they think about valves on sticks, and they think about medicine in its biggest picture, as it were. So I'll ask you the same question again. Do you think that cardiovascular medicine is ready for a personalized approach?
I think that is ready in the sense that there is a lot of work in that direction. I say again that definitely is clear and evident that in not most of the thing we do every day in our words and in our clinics is personalized. But I think that the promise for a broader approach is there. The important thing is that clinicians, no matter what they do, they start understanding and working in the direction of trying to identify how the patients can be different from one another, rather than keeping with the tendency to say they are all the same and they get the same approach. That even not in molecular, doesn't have to be only molecular, the personalization of medicine.
Absolutely. So we keep fighting the good fight, yeah?
Mm-hmm.
Okay.
Yes.
Filippo, perhaps I can hand back to you.
Silvia, thank you again for joining us for this very interesting dialogue. And I'd like to change page. The European Heart Journal, we pay considerable attention to sex and gender issue. For instance, with Yolande Appelman and Roxana Meran, we've recently published a viewpoint on equity, gender and sex equity in trials. Do you think that this is an issue, sex and gender equity in trials, something we have to improve?
Oh yes, for sure. You know, it has been now many years that the point of the need to understand better the diversity of the clinical manifestation, the age profile, and you know, basically the gender modulation of the cardiovascular diseases. First, ischemic heart disease itself. So as we are saying that not only gender is one of the issues where we need to pay attention, but the age of the patient, the ethnicity of the patient. So there is a lot in which we can improve in the design of the trials. We have taken the easiest way, that is have a predominance of males and usually white males, you know, in our series of patients, and is now the time that is thinking about personalized medicine. We try to get cohorts also of patients that are different than the standard group that has been involved in the trials.
Are you aware of initiatives at ESC in this regard in order to promote equity in trials? Do you have sessions this year or something in perspective?
I think that the issue of gender is every year in the Congress is touched under a different perspective. That can be drug metabolism, that can be epidemiological profile, and so on. I cannot really spot a specific session on the need to do more efforts in that respect, but definitely is a topic that is on the list of the issues that are considered worth discussion. And with that, it also go the professional component, you know, that is that the involvement of women in management of cardiovascular diseases, that is still a sensitive topic, especially in the invasive component of medicine.
We are in the COVID era. Let's come to COVID for a while. There are several signals that women are paid more than men during this COVID pandemic. For instance, the proportion of manuscripts submitted by women is dropping. Do you have the same impression? Are women paying more than men in this setting?
Well, when there is a social stress, very often women, for several reasons, get involved to a higher level. So if we think that a big proportion of the population has remained at home for a period of time, then home is the women place, you know, so all of a sudden many women have found themselves at home having to struggle the smart working that is far less smart if then at the same time it means that you have to take care of the children of the home and manage the safety of everyone in the family. So to which extent this has affected the women cardiologists, it's maybe harder to identify, but even living at home, a portion of the family that no longer can go to kindergarten or to school, or the husband that is at home with the children that are not going to school and kindergarten, you know, it creates an anxiety for the person that feels responsible for that. So I think that this is evident also from data from the increase of requests of psychological and psychiatric help, that I've seen some statistics supporting the fact that women were a higher prevalence into those as well.
So regardless of COVID, you think that in a broader perspective, women are penalized in this in the scientific career and in the scientific societies? And if so, you see step forward in improving this situation?
Well, I think that for a woman, it's always more complicated, and it is more complicated also from an educational point of view, that one meaning a woman, you know, tends to feel responsible for the well-being of the family to a larger extent, tends to feel guilty of the problems that may develop within the family. So to find setting and conditions that allow the equal perspective and possibility in advancing in the career are heavily influenced also by the socioeconomical status of the women. So I think that is a more complex situation that creates an inequality within the inequality, because definitely women with a better social status, more advanced, more money, can solve the problem or address the problem of child care, even when in countries where the state is not providing support for that, but hiring personnel and to help. And women that don't have that clearly are in a much larger difficulty. The closeness of the family, I mean physical distance, how close the parents are, how well they are in providing support, are also social factors that may deeply influence the possibility of pursuing a complex career like one in science and an academic with an academic perspective.
I would like to remind all of you that this dialogue is a live stream and that you can ask questions. And indeed, I'm reading a question coming from a colleague from the University of Malaya. It's a rather broad question, but I find it interesting. What about lifestyle and sudden cardiac death? How can we minimize the risk of sudden cardiac death by improving our lifestyle?
Yeah, it's a complex question in the sense that you can approach it from different perspectives. Lifestyle includes a lot of factors. So the first that comes to mind in cardiology, of course, is the diet as a factor that influences, the probability of ischemic heart disease, that at the end is the first cause of sudden cardiac death. Then we can see also, we can jump from physical needs like eating to the psychological perspective and see how stress, and for example, the stress that we are seeing in the COVID period, can be a factor that influences the susceptibility to cardiac arrhythmia. So indeed, lifestyle can also be protective. And so, people put exercise in their lifestyle, they can reduce the risk of sudden cardiac death. So definitely, sometimes it's hard to quantify in an individual who has a cardiac arrest, which percentage of the risk came from the lifestyle of the person, but definitely the evidence of the role of the single factors, in normal individuals that don't have a lot of risk factor identifiable at a clinical visit, all the way to the spectrum of the patients with inherited diseases, like, for example, right ventricular cardiomyopathy, where it's well known that exercise is detrimental. I think that this interaction between substrate and lifestyle is very intriguing, very interesting, and not fully disclosed and exploited in the management of the patients.
Terry, I'm sure you have many more interesting questions, please.
Silvia, speaking of my own field, I think in the past 1 to 2 years, the whole therapeutic landscape has opened up. I think I'm having more conversations now about new therapies, whether they be repurposing of old drugs, small molecule therapies, or various ways of altering gene expression. Do you want to maybe talk a little bit about the work that you're doing now and how you see this panning out for inherited arrhythmia over the next 5-10 years?
Yeah, you are right on the spot. I don't know what has triggered this interest in cardiovascular medicine from the point of view of development of drugs and therapies in general for unconventional patients. But in parallel with the fact that the several pharma companies have discontinued their interest and commitment to cardiovascular, it is very clear the signal that on the other side, biotech and companies interested in rare diseases have increased a lot, their interest. And you're right that your field, hypertrophic cardiomyopathy, has been sort of the pioneering one, probably because the disease with this prevalence of 1 in 500, that is the magic number that everybody refers, seems to be attractive by being a good balance between an uncommonly considered disease for a trial and a disease that is common enough to allow the enrollment in the trial. That is true that this energy that has started with the hypertrophic cardiomyopathy is now extending to other fields of the rare disease in cardiovascular. And so I think that is actually also for the perspective of a scientific society, I think that the more contact with biotechs, not only contact with pharma that is at the most advanced lab phase of development of the drugs, could be very interesting and beneficial, because it seems that more and more are these companies that start raising money with crowdfunding or investor funding and dedicate to these specific topics that may lead to the development of novel therapies, but drastically novel, you know, with different approaches, different class of drugs, and maybe are the most exciting, definitely for me, are the most exciting side of the new development in our field.
Absolutely. I mean, I think one of the biggest drivers for change in medicine is the development of new therapy. It's tell me about a disease when I can do something about it, sort of thing. And I think the poster child for that is very much amyloidosis. Amyloidosis has always been there, and people weren't interested in it. But all of a sudden, there's now a therapy, and we're diagnosing it, and we're now treating it. I mean, what's the analogy for inherited arrhythmia? Where do you think we're going to be starting first with molecular therapies in inherited arrhythmias?
So I think that repurposing of drugs is an interesting perspective. We have started from the repurposing of mexiletine, that is a partial repurposing because it was an antiarrhythmic, but with long QT3 becomes a QT modulator. And so that has been a signal that we could have molecules with electrophysiological properties that can be beneficial. Then, we have some other targets, for example, with patients with a short QT, the use of drugs that prolong the QT has been attempted, and it seems to work, at least to reduce the probability of a sudden cardiac death in this population. So I think that understanding better the electrophysiological component of different compounds, can open a promising perspective. The one in which we are most interested, however, is the attempt to modulate the genetic substrate. The heart not being a replicating tissue, the cardiac tissue not being replicating, has more difficulties in the landscape of gene editing and base editing, because those possibility of repair in the DNA sequence occur mainly during the division of the cells. But still, also in that field, there is a lot of research and a lot of efforts to develop. And then there are the gene therapy to fill the gap of the loss of function mutation, haploinsufficiency, genetic defects, that there are still some obstacles in the sense of efficiency of the infection of the heart cells, identification of promoters, identification of capsids. But it's exciting to see at the level of the biotech how much efforts and investment are being dedicated to this. So it's a great privilege to be in that journey at the moment.
Absolutely, as you say. I mean, one of the big challenges is getting the gene into the heart, isn't it, at the moment? And there's all sorts of very interesting and some very scary approaches being talked about at the moment. But do you think we could be looking at cure? Do you think you could imagine that we could cure diseases? I mean, I sometimes find that a little difficult with cardiomyopathies, particularly to advanced disease, but you could see perhaps a cure for long QT syndrome or a cure for CPVT.
I think that what is still early to say is the balance between fixing the defect and being sure that over a very long term, because it's going to be a lifetime therapy that is always there and that you cannot turn off, at least at the moment, that there is to be a balance between the potential toxicity. What is difficult in my mind to think, is whether when the heart will age, age as a change that occurs in everyone, what would happen in this production of a protein intended to compensate a lack of effectiveness, but maybe then it can become toxic over time when the substrate changes. So I think that this answer will come only when the trials will be done, and we will discover by doing, as medicine always work, that the long-term effects are only known when you have patients treated for over the long term. But it is exciting because we know that a lot of patients, for example, in cardiomyopathies and cardiac arrhythmias, fail on the therapy because of compliance. So the big promise of gene therapy could be the fact that it doesn't rely anymore on a patient that is compliant and takes a medication every day, multiple times a day for the rest of his life.
Yeah. Do you foresee any ethical or social issues related to gene therapy? And I suppose what I'm thinking about here is the fact that these therapies are going to be expensive. And talking now about one-off cost of maybe €1 million or €1.5 million for gene therapy, and of course what that means is that that's going to be prohibitively expensive for many healthcare systems. So there's going to have to be choices made about what gene do we treat, who gets the treatment. Are these things that you're starting to think about now?
Yeah, I think that in that respect, the answer that I'm getting from people in the field on the side of pharma and biotech is the fact that we had to make this shift from the difference that exists between preparing drugs that are chemical compounds, where we are very well structured to produce, maintain, distribute, and all that, these compounds, and having a live element that is your therapy, and so can survive, can die, can degrade. And that the answer seems to be that the changing scale in the bioreactors, the larger bioreactor that can provide, of course, a reduction in the cost because you have a larger scale, seems to be the answer, or at least the answer in which most of the companies involved in the gene therapy are betting on. Of course, to have a larger scale effort to reduce the cost, you need to have multiple gene therapy products on the market so that then you exploit the production of the viruses. So at least an answer in how to reduce the prices seems to be coming, and that of course is necessary because no one could afford the drugs that cost €1 million per person to treat, unless the cost for having this person in the hospital every month or so, as it happens for some metabolic disease, coagulation diseases, make it worth. But in the cardiac field, I don't think that we are in a situation where the patients have this outrageously expensive care if they don't have a cure like the gene therapy. So for us, it would really mean to have a reduction, a significant reduction in the cost of the therapy.
Yeah. And finally, if I may, just to come back to my theme about, is cardiology ready for personalized medicine? So, if we're going to get into this field as a specialty, we're going to need at least some of us who understand genomics. Mm-hmm. Because it's going to be the genomic cardiologist of the future who's going to be selecting patients for these new drugs, new gene therapies, monitoring their safety and efficacy. So what do you think we have to do as a community to raise the level of genetic literacy amongst cardiologists, cardiovascular specialists?
Yeah, it's becoming a specialty. It's already a subspecialty of medicine. And I'm seeing this very clearly as I'm working with some companies in thinking of clinical trials for different of these inherited conditions. And invariably, it comes to the interpretation of the mutation. In whatever disease you have, is this a true mutation? How we want to enroll patients that have a mutation that we want to fix and correct. So we need to be sure that we are not enrolling also patients that have variants of unknown significance or so. So who decide and how we build the consensus of that is something that I am frequently addressing, with the partner from the industry side, because they are not structured and we are not structured yet for getting the precision in the interpretation of the genetic defects that is required when you need to randomize your patients for the treatment. So yes, we will have to learn more. We will have to put also the field of interpreting the mutation, not just by in silico means, but developing a assay for the interpretation. In ion channels, we are lucky because with patch clamp, we can get quite good insight, but for the cardiomyopathy, we don't have that. So the development of assay to define what is the true consequence of a change in base or truncations or whatever, you know, in the mutation. It's clear, for example, very much with the ryanidine receptor. I always go back to my
your favorite receptor.
Exactly. But now it's becoming a very complex field, and now there has been the discovery that loss of function mutations are there. So we used to say it's a gain of function unless it's considered a variant of unknown significance or a benign one. But now, you know, we have something that can be, from an in silico perspective, a damaging change, but it may be damaging in the opposite way. So a loss of function rather than a gain of function. And we don't have a say to understand it, because the expression of the ryanidine receptor in heterologous cells is not an easy thing, you know, and is expensive itself. So yeah, there is to develop, there is the need to get a higher level of precision in the interpretation through wet biology, I guess, not only try to get the answer from in silico biology.
Yeah, and I think also I think we should not undervalue the contribution that specialist clinical genetic cardiologists, if you like, can make to that field, because ultimately it's got to come down to a human phenotype, isn't it? So, I mean, you know very well from the work that you've done over the years that sometimes what happens in a dish doesn't always translate into the human, or at least not in the same way as you perhaps anticipated. So I think the human phenotyping is going to be critical to this problem of interpretation, I think.
Yeah, absolutely. You know, and we have now a pipeline that is growing in the scale of the model. You know, the IPS cardiac myocytes are becoming already, after few years, you know, something that, yeah, okay, but it's not enough. We need the organoid, and then we need the organoid on a chip, and then we need the heart in a dish. And so that is something that is quite promising. Still, you know, the safety can be assessed only in organisms. So I think that the animal models will remain an important component to integrate the toxicity in multiple organ systems. And so probably the cost of development is not likely to go down, you know, for these therapies. You know, if you think of the complexity that starts with the interpretation of the mutation.
Okay, so basically we need a new subspecialty, genetic cardiology, yeah, become the largest association within the European Society of Cardiology, perhaps in 10 years' time.
Well, I would say that is already there. It's not only cardiogenetics cardiologists, but also genetic counselors that are fully dedicated to cardiac diseases. So I think that the specialty there is not formalized yet, but is the profession is lived with a subspecialty in cardiogenetics.
Yep. Great, thank you, Silvia. Back to you, Filippo.
Well, Perry, I have an answer for you. We already generate a lot of interest in the field of genetic medicine and personalized medicine, because I'm receiving many questions for you, which means that you're raising the interest in this field, and not surprised, because we are two main stars, Silvia in the field of arrhythmias and you in the field of cardiomyopathies. And indeed, I have a question for you, Perry. Colleague Stein Reinstein from Brazil. What is new in risk stratification of hypertrophic cardiomyopathy?
What is new? I think so. Let me just step back with risk stratification. So one of the biggest fears that people have with hypertrophic cardiomyopathy is the problem of sudden cardiac death, and that is completely natural. You know, I say to all my patients when I see them for the first time, you know, when you're told by a doctor that you have hypertrophic cardiomyopathy, you go home and you have what I call a Google moment. Because if you put those two words into a search engine, all you see is sudden death, sudden death, sudden death. But of course, the first message about sudden death in hypertrophic cardiomyopathy is that it's a rare event. You know, the overwhelming majority of people with hypertrophic cardiomyopathy never have sustained ventricular arrhythmia or disapnea. And actually, the big problem in hypertrophic cardiomyopathy over time is heart failure. It's heart failure and atrial fibrillation. But of course, it's that rarity that makes it hard to predict. And the approach that we've evolved over the years is to use various clinical markers that associate with risk. And again, homologists will argue about their favorite biomarker in a bar. I believe it's non-sustained VT, I think it's left ventricular hypertrophy, I think it's an apical aneurysm. And again, all this stuff is telling you is how bad your heart is. So if you've got a lot of hypertrophy, a lot of scar, a lot of ischemia, you're going to have some non-sustained VT. If you've got obstruction, you're going to black out. Yeah, the more of this stuff you have, the worse is the underlying substrate. Actually, I think if you follow a systematic approach using the approach that's been developed over the past 20 years, looking at features that associate with risk, you can identify the overwhelming majority of individuals who are at risk of dying suddenly. It's a really rare event now. If I do my retrospective, the one I was asking Silvia to do at the beginning, when I started out in the early 90s in hypertrophic cardiomyopathy, we would be getting phone calls all the time about someone who dropped dead suddenly. It doesn't happen to me anymore. And it's not because they've all got defibrillators. We've identified those at risk. We're managing the disease. It's about global disease management, preventing stroke, maybe even treating some of the heart failure. But I think we're trying to improve all the time, but actually we're pretty darn good at identifying the high-risk patient. What we're not very good at is treating the basic underlying muscle problem. We don't have any therapies that are known to reduce the rate of remodeling in HCM. So sudden death, what's new? There's not a great deal that's new, but what we do is pretty good. But what we really need are advances in heart failure.
Well, thank you on behalf of our colleague for your exhaustive answer. Now, Silvia, we have a question for you from Igri Rao. Can you comment on the risk of sudden death after COVID? We have data on sudden death in patients who recovered from COVID.
Yeah, we have been trying to look at the occurrence of sudden cardiac death in COVID, and as many other groups have tried, we all had these patients to follow, and especially after COVID to see their profile and the consequences of myocarditis. So it is pretty documented at this point that the consequence of myocarditis can be leaving the heart with scars. And something that is maybe less expected is the fact that athletes and individuals with no cardiac diseases, and even the one that didn't have a COVID with a lot of symptoms could be identified as having scars as a consequence of an undiagnosed myocarditis. So that is one step, one point. The other point is that we have had in our cohort of patients with inherited diseases the co-occurrence of the presence of the disease and the occurrence of a sudden cardiac death episodes. And it is, and at the time after the patients had COVID or was still affected by COVID. So it's difficult to say if it was a coincidence or a cause-effect result. In some of the patients, especially the one with the channelopathies, if you all of a sudden find scar and the patient has had COVID and the first cardiac arrest or one recurrence after a long time from the previous one, you may suspect the relationship with the COVID infections. In patients with cardiomyopathy, it's more difficult because they already had pre-existing scar. But the topic is it's interesting. I have not seen large series of patients from the genetic world, from patients with genetic diseases, and whether in this group the occurrence of sudden cardiac death has been higher. But it seems that it's not trivial the number of people that have had a cardiac arrest or an arrhythmic recurrence leading to syncopal VTs after the occurrence of COVID. So I think that at the beginning, we were all underestimating the cardiac component of the COVID, maybe not the cardiologists, but the community in general was so focused on the lung. And now it seems that the lung recovers because it's a tissue that can replicate, and therefore the long-term consequences are more in the heart, that when replacement of fibrotic tissues occur, of course, create a predisposition to arrhythmias that remains for a long time in the so-called part of the syndrome of the so-called long COVID.
Now we have a question from Carlo Patrono. What about incorporating personalized medicine, this paradigm in guidelines? Are we there already or sometime in the future? Challenging question.
Yeah, it's an interesting question because it leads to try to assess and distinguish between the things that are preliminary evident and the one that are mature for applicability in the clinics. So I think that the guidelines are trying to exploit, every time there is the possibility of going to a subtype of a disease approach and say in this subset of patients, and this has occurred in the last several years. For example, just think about the laminase C mutations, that has become really as a type of cardiomyopathy to be treated with a focus on the defibrillator. So whenever we have a hint, in introducing in the clinics, I think that when there is a sufficient evidence, there is a great attention to introduce it. The problem is that it's not so big and so impactful that we could really say we have now a personalized approach for most or many of the cardiac diseases. But definitely we shouldn't miss the opportunity to introduce any evidence supported by data that we could treat the patients in a personalized way.
Yeah. Thank you, Silvia. And now I have a question for both of you. A bit of competition. As I said before, you are the champions of personalized medicine in arrhythmias and cardiomyopathy. Which field, in your opinion, is more advanced in regard to personalized medicine? Arrhythmias or cardiomyopathies? Silvia first.
From my perspective, as I said at the beginning, is long QT syndrome is the winner. And, it's just, I don't know if it's really because there has been a lot of research in that, but I doubt. I think that just it happens to be a disease with the right number of few genes, mutation that can be characterized very well in vitro, ion channels that have been very well known at variance with many of the protein of cardiomyopathies. Who has ever studied filament C before the identification of the mutation? So there is a lot of basic science understanding that is still needed in the cardiomyopathies for their complexity. So I think that if we only could move the other channelopathies to the level of long QT syndrome, but sometimes it's really the unknown that are still big. Like in Brugada, we have had up to 20 genes, and now we are demoting most of them and saying that maybe the only gene is SCN5A, the sodium channel. So all this uncertainty on the basic science is still limiting the expansions of the conditions, inherited conditions in which we could claim that we have a truly broad and impactful personalized approach. And I think the long QT syndrome remains the winner.
Perry, what is the winner in your field?
The winner, for now,
it's
long QT syndrome. But I think things are changing very quickly. And if I look at the cardiomyopathy field, I think dilated cardiomyopathy is where a lot of the action is going to be. It already is there. The notion that dilated cardiomyopathy is an idiopathic disease is 1980s medicine. If you look hard enough, you can find an etiology in dilated cardiomyopathy. Minimum 1 in 4 will have a genetic etiology, a definite genetic etiology, monogenic disease. Others will have inflammatory disease. There'll be a few infiltrative patients in there. And one of the big advances we've had with our ability to perform genetic testing in the clinic now is that we can start to stratify dilated cardiomyopathy in exactly the same way as oncologists do, say, with lung cancer. Oncologists don't talk about lung cancer anymore. They talk about 70 different diseases. Well, we're moving in that way now in dilated cardiomyopathy, where it's whether it's filament, whether it's lamin, whether it's desmoplakin, whether it's desmogliin, whether it's desmin, put all those things together, you're actually talking about a lot of patients with diseases that behave in totally different ways in terms of their arrhythmic risk, their rate of progression to heart failure, And already we're starting to see signals that they respond differently to conventional anti-failure therapy. So I think long QT syndrome is a beautiful model for genetic disease. As you say, it's a relatively small number of genes. I think the pathophysiology is well understood. The targets are very tractable in terms of the drugs that we have available to us and other interventions. And I think it's going to be more complex in heart muscle disease. But it's, I think we're going to give long QT a run for its money.
Very, very interesting. And now, Silvia, from science to your biography, your career has been remarkably transatlantic. And this is the beauty of science. We are part of a global community, and this makes our job particularly fascinating. Based on this transatlantic experience, what are your suggestions for our younger colleagues?
I think that the suggestion is to introduce in their training periods abroad, but not only when you are in the early phase of training, going abroad, visiting another clinical center, another research laboratory is stimulating and regenerating at every point of your life. So it's a fantastic experience to be in the fifth decade of your life and say, okay, now I'm starting a new lab in this center because it's a fantastic center. And this is what I did 5 years ago when I decided to start in Madrid, that being in front of the bench empty and saying, I have to purchase the equipment, I have to select the people, is something you do when you're 30, not when you are my age. But still, I want to do it because it's really regenerating. You put yourself under question, you ask, am I following the development? Am I advancing my approach to the diseases? And this is necessary, and nothing like the exposure to a new environment, a new language, a new everything. It resets your brain to be ready for a new challenge, and is really rejuvenating from a mental perspective. So I would recommend them, never say I am too old to take 1 year off and go somewhere else to learn.
Thank you, Silvia. Well, you are not only a top scientist, but also the vice president of the European Society of Cardiology, an important position. How do you envision the future of the European Society of Cardiology? What is the experience?
Well, I think that what has happened in these last 2 years that has placed the society under the need to reassess our policies, our strategies, our source of revenues. It's something very healthy for a society. It's similar to the process that I have just mentioned for, when you decide, okay, I go into a new land to start new approaches, I learn as if I were 20 years younger. The same has occurred for the society in this time, where we have started looking at the potential and the limitations of online approaches for education, for example, for Congress, and this also for a society means deciding which are the source of revenues, and the source of revenues strongly influences the structure of the society, its internal organization, the process for developing the strategy. So the society has embarked in a very important process driven by the presidential trio of reassessing, what should be the strategy for the future, and taking from multiple perspectives the educational role of the society, the advocacy role of the society, that in the last few years has really grown and provided new opportunities for speaking with patients and listening to patients. And so I think that it's a time that is really an energetic one, and with a lot of potential for designing on this successful model that the ESC has developed over the years, that has been named the European Society of Cardiology, but being recognized by the physician as a worldwide society of cardiology. into a new model that can encompass other activities like the one of patients, novel modalities for disseminating education, monitoring the competence of physicians by developing tools for evaluation of competence of the clinicians. And all this is being a part that requires a lot of energy, but I see a lot of enthusiasm in the ESC leadership toward this opportunity of redefining the strategy of the society for the future and maybe its mission as well.
Silvia, I'm overwhelmed by questions coming from the audience. Well, just to finish, one is from Nikos in Athens, and the question is about the role of genes in ischemic heart disease. Do you think that polygenic risk score will improve our risk prediction in ischemic heart disease?
Well, I think that ischemic heart disease is getting there at quite a rapid pace. You know, there are several of the polymorphisms that compose the risk score on chromosome 9, for example, that are actually already demonstrated to be stronger than some of the biological risk stratifiers, like lipids, for example. And so I think that it will be quite soon that the genetic component will be part of the risk score charts for ischemic heart disease.
And the last question from the audience is about artificial intelligence. Any role for AI in the prediction of sudden death in your crystal ball?
Yeah, well, when something becomes so mentioned, so much attempted with data that seems miracles, but then maybe they don't get applied to the clinic after 3 or 4 years, that has already occurred, especially in some of the arrhythmic studies. You wonder, you know, whether the time is ripe now, but definitely looking at the perspective, it will come, it will become it will develop a solid approach, methodological approach, the definition of the size of the samples required, because some studies are clearly underpowered at the present time in the absence of solid methodological rules and approaches. It is very scary when I'm thinking from the perspective of the physician, the fact that you know that there is a computer that can tell you this will have sudden death, this will not have sudden death, and you don't understand why. You know, you want to stand in front of this computer and tell, yes, but tell me why. We are not used to act on a risk stratifier element without knowing why. And I am not sure if I have to think with concern or with relief to the fact that one day, forget it what it is, you know, it's just tell me that this patient has a problem. But I feel that I will never love medicine without the understanding of the underpinning of a medical decision. So I think that it will come, it will produce results, but the challenge will be to get out of that computer the answer of what is that the computer has identified, the human being have not identified.
Brilliant answer. Thank you, Silvia. Before my last question, Perry, do you have any last question for Silvia at the end of this fascinating dialogue?
I suppose, what's the next 10 years hold for channelopathies, Silvia? I mean, we've touched on a lot of areas, but if you were going to, what would you like to see, your top 3 sort of successes?
Well, first of all,
you're not allowed to mention the... Sorry, I was going to say, you're not allowed to mention the ranitidine receptor, okay? So.
No, absolutely. Not only, not only that. No, I think that I would spend my next 10 years in science developing a gene therapy. This is my commitment, and my group is enthusiastic about it. So I know that we will go for that. But there will be other aspects that I think can be really interesting. So the collection of large databases, the merging of databases, the collaboration at international level really holds the promises of understanding better and in a more analytical way the phenotype of the patient, that sometimes we tend to freeze in few markers and then say that's all, we know the entire story. So it seems old-fashioned as compared to gene therapy, but I think that deeper phenotyping, with the technology or just even molecular biology, pathology, proteomic applied to tissue of human being, these are things that we are already seeing that in the animal model, proteomic can tell us a lot. And so that could be a good component, especially for the heart, where we can get biopsy after all. So that is the second part. The third part, what I wish will happen is in my field, the fight to sudden death, an improvement, but really a quantum leap change in the technology for defibrillating the heart. I'm quite disappointed that the field, both on the medical component and the industry component, has relaxed and said we have the defibrillators. We have the defibrillators, but we don't have the super duper defibrillator that technologically we could have at this point. And so we cannot just accept that we can stop here and say, yes, they are big, they give inappropriate shocks, the catheters breaks, but it's good enough. There is no good enough when you have to treat the patient. So that is the third thing that I would like to see, and I hope that will occur in the next 10 years.
Fantastic, thank you.
Silvia, this is my last question. The European Heart Journal dialogues have been very successful, but how can we improve further this success? What are your suggestions for the dialogues?
The dialogues are very nice. I think that is nice, the balance that you are able to keep in mixing the professional component and the personal component. With your questions, you bring out the personality of the speakers. And so this is something that as a person in the field, I like very much to understand and discover with you as you ask the questions. In that respect, probably to find a way to have 2 or 3 cardiologists, not super expert, but just 2 or 3 cardiologists interested in a topic who ask questions, and can be the question that come through the chat now, but could be asked by young cardiologists, for example, could be an innovation that maybe adds another component, and giving the opportunity to interview the expert in a field to young people might be refreshing and provide surprises in the type of question that they may have from their perspective.
Brilliant. Thank you, Silvia. This is really an excellent suggestion, and we'll work on that. So this was my final question. First, let me thank Perry Elliott and Silvia Priori for this wonderful hour, this wonderful dialogue. And now I have an important announcement for all of you, because our next dialogue, which will be on August 26th at 17 CET, will not be a dialogue, but will be the editorial board of the European Heart Journal. Every year, we have the editorial board of the European Heart Journal at the time of the Congress, annual Congress of the European Society of Cardiology. Well, this year, the editorial board will be virtual and will be open to all of you. So you're all very welcome on Thursday, 26th of August at 17 CET. Perry and Silvia, thank you again for this wonderful conversation. Bye-bye.
Thank you for having me here. Thank you very much to both of you.